Right, can you hear me okay? Excellent. Well, thank you for the introduction. Thank you for inviting me. I'm a surgeon. Remember that when you're asking questions. And a surgical trialist as well, I should say. So thanks to Babak for inviting me to talk about Rossini. We're going to talk about Rossini 2 mainly, which you've already heard a bit about, but a bit of Rossini 1 and why it was so inefficient and why we came to this design. And then a little bit later on about some of the more progressive things we are currently planning with this wonderful MAMS design. So it's all about wound infection. Clinical application here, SSI or surgical site infection is wound infection to most of the public. It's now the commonest complication after surgery. So we've spent lots of money and time on MRSA and C. diff and catheter-associated infections, central line infections. Not a lot of money and time on wound infections yet. And it affects an unacceptably high proportion of all operations, wherever in the body. In my part of the world, which is abdominal surgery, it's about 1/5 to 1/6 of elective operations and maybe 1/3 of emergency operations. It has a really high impact on patients. There's a horrible picture there, which I'll take down in a second, but it's to remind me to mention that it's not all just antibiotics and a couple of dressings at home, as some are that you may have encountered in loved ones. It can be a devastating return to theatre, ITU, and even death from a wound infection. And we think the average cost per SSI is about £4,000. If you do the maths, it's about £1 billion a year we are wasting. And I use that word carefully because this is probably preventable. There's lots of things we try and do as surgeons before the operation, in the operation, and afterwards, but most have really poor evidence, as we'll come back to later. We are learning that most wound infections are multifactorial in etiology, and therefore a single magic bullet to fix it, one new stitch, one new device, is probably wishful thinking. And as I've said, in these current times, 7 million undone operations in this country, unprecedented pressure. Having this complication, I think, is pretty unacceptable. So another nasty picture, sorry about that. You didn't expect that at this meeting. Rossini was a trial that we designed back in 2008 when I was a registrar. We had our new research collaborative formed. That's a different topic which we won't go into today, but we wanted a really simple intervention. We had one surgeon in Walsall, very small hospital in the West Midlands, who had really low wound infection rates. And all of us who were registrars spotted this and we worked for him. And he did a lot of things, one of which was to use this pretty old device, which is a plastic ring. You put into the wound, you unfold a kind of bin liner, and you operate through it. And we thought we should do a really simple trial, randomizing patients to get it or to not get it. 1-to-1, really simple. It was funded by RFPB. It was one of the first full RCTs they funded to the tune of about £250,000. And it did well. It published in the BMJ. They don't work, by the way. When we were planning a follow-on study, this is partly us thinking what to do next, but it's actually also us doing that thing that all normal trialists do. You spend ages thinking of a design, getting it funded, getting it up and running the trial, and then spending some months waiting for your data cleaning and your analysis to come through to see what the result was, only to have the inevitable, negative. And then it was around this time, 2 things happened. I had this minor epiphany that we know the answer within 30 days. By definition, literally the definition of a wound infection is within 30 days, was there a wound infection or not? So to wait 3-4 years, as all our trials were doing, was bonkers. I then also heard some talks by Max and Matt Sides and James Wasson about MAMS design, and Stampede was really up and running massively at this stage, and we thought maybe we can do something here. So exploiting that 30-day outcome measure, the fact that you know the answer 30 days after your intervention's applied, guaranteed, would allow us to do something more efficient, to look at multiple arms simultaneously, to drop arms that weren't working. And we also did some work looking at what we do, and I said earlier there's lots of things we do. It's about 60 variabilities that we use. And we set some rules and we said, well, let's look at multiple ones. Going back to that theory that one magic bullet was unlikely to be useful, let's look at 3 things we do alone and in combination. And that's where the desire came from. The rules we set for Rossini 2 were that they had to be kind of biologically plausible that they might work together. So we couldn't pick 3 really similar things at the same point of the operation that would cancel each other out. We had to have things that were available in the NHS but not in abdominal surgery. For example, standard of practice in cardiac surgery but not in the abdomen, that would be perfect. I had to have a CE mark, so it had to be available in the UK market. We couldn't do a CTIMP trial as well. So I had to have a CE mark, and we wanted them to be roughly the same price as each other to help with the health economics. And like I said, we wanted to look at how they worked in combination. These are the 3 we picked. And we went through the HTA process of evidence generation and proper reviews the kind of signal that they might work, but they weren't standard practice at that point. A skin prep compared to standard other stuff to wash the abdomen before surgery. This incised drape, which has got iodine impregnated into it, and you cut through it, and it sticks the bugs in place, allegedly. And then at the other end of the operation, a sponge that dissolves away, made from bovine collagen, that delivers antibiotics into the wound, not systemically, but into the wound where the bugs are, we think. Yeah. And they were the three we picked. That's the design. You've seen a picture of this from Babak just now. I prefer his. I'm going to get a copy of that slide from you. I don't know how you did it. It's beautiful. This is our older version that I made in, I think, PowerPoint 10 years ago. A control arm, which is double randomized to where they get nothing. Arms B, C, and D, where they get one intervention. E, F, and G, where they get two interventions. And H, where they get all three interventions. You can see there's a pilot phase, as per any HTA-funded trial, where we said we'd just check it was working, check we could centralize the data fairly efficiently, because that was a necessary point, and that had never been done in a surgical trial till this point. And look at it, compliance and whether all the systems worked. And we added in a second internal pilot, if necessary, which is a neat trick that I've done all my HTA trials since then, if you don't meet your traffic light greens. We then had our interim analyses. I'm very grateful to Gordana at first, and now Babak for helping us with all of these complex maths that you've seen as to when we have to break the code. And with this went to the funder 3 times before we got it funded. The first time they just said it wasn't efficient. And it turns out they wanted efficient, they wanted some other form of efficiency in the mid kind of teens to do with the routinely collected data. The second time, we went with a normal MAMS when any arm could have continued as long as it wanted. And they didn't like that. And some of the feedback alluded to, and we eventually realized they didn't like the blank check nature, because we couldn't tell them how long it would take and how much money we needed. And then we went back with this drop the loser design, proper name selection design, which you've heard about from both speakers so far, where we said no matter what, we will drop 2 arms, the worst 2 arms we will drop at the first interim analysis, and then we'll drop 2 more minimum at the second interim analysis. So that allows you to know how many patients you need, therefore how much money you need, because you can predict how long it'll take. And it was that change, I think, that made them fund it. So that's a kind of practical example of what Babak just mentioned. Just in brief, because I'm talking about a trial, we made it really pragmatic on the pragmatic explanatory continuum, pre-C2 and all that wonderful stuff. This is right at the pragmatic end. So any abdominal operation, any indication, any surgeon. And because it's so big, we can kind of cope with some of this dirty water in the analyses. We've used the same inclusion-exclusion criteria in all of our wounds trials. That allows us to pool the results, by the way. Primary endpoint and secondary, as you'd expect. So an update in brief, you can see the flat point on March the 20th, 2020, when all the trials shut down in the UK pretty much. You can see that it picked up slowly after the pandemic, the red line. Another flat point at each national lockdown. You can almost chart them on here by date. But as of last week, we're at 4,650 patients, if not a few more now. So we're getting towards the end. You can't read that from the back. That's just to remind me, there are 53 sites, of which 51 are open, and now 50 are actively recruiting, which I'll come back to in a bit. First analysis, I'm not going to have any complex maths on here, but it can be summarized as that. So you'll remember the maths, that was in 4 arms, not just 2 arms. It robustly didn't work. So actually all 4 arms of that were taken out. So we were left with a slightly sadder looking than predicted diagram. You can see the control arm versus 3 interventional arms. The second interim analysis is that. So we got rid of both of the arms. That also doesn't work. And we're left with this really sad looking current stage of the trial now. So control versus skin prep is where we are right now. And of course that means we are in the final phase of the current trial. But we think we'll get that finished by September, October. In fact, we had a really good month. April was awful with the doctor strikes and Easter. May was a fantastic month in terms of recruitment. We've gone back to the funder and we've looked at the first 4,000 patients. Our control arm rate is higher than we thought. It was funded at a 15% baseline rate. We've got a large number of elective operations and keyhole surgery operations, which any surgeon will tell you has a much lower infection rate than open ones. It's just not true. 2/3 of ours were keyhole. So this can be summarized as we haven't solved the problem yet. We very definitely haven't solved the problem with what we've done so far. But we've got a really efficient system. We've got that football stadium, which I love that analogy, up and running with 50 sites actively interested, and every time we drop arms, the recruitment increases because everyone's interested in things. And we've gone back to that group and said, if we could add new interventions in, what would you like to look at? If there really was a possibility. There's a couple of things that have happened in the past 2 years. This trial has come out from our colleagues next door in Birmingham on the global surgery unit around glove and instrument change, a cluster randomized trial in the Lancet. You may have seen it. This robustly works, which is nice to find something that does. And this is becoming, people are talking about this as becoming standard practice internationally. It's almost free, apart from the cost of gloves. You can do the instruments just by changing the way you actually operate. And this has come out from a fancy wound dressing that has got nice recommendation to use in vascular and cesarean sections. It's supposedly cost-effective and clinically effective. It sucks the bacteria into the dressing and binds them there. I'm as cynical about it as you are from that description, but it needs careful assessment. So we've said to the HTA, why don't we do this? Our final analysis is that thick dotted line, and it's going to be this autumn for sure, looking at the numbers. Wouldn't it be nice to take the results and make the new skin prep, if we've proved it works, our current standardized care across the trial, if you like our control arm practice, but actually across all the arms, not just control, and look at, in the existing framework, 2 new interventions in this way. And we've said, this would be efficient because of the football stadium thing. If any of you have costed a trial, just look at the bottom one, which I think is the more likely one. We can do this. We can add 4,200 patients, 26 more months to the existing trial structure for only £600,000. If that was a new trial being proposed to the HTA, it'd be 2 million, if not more. So it's incredibly efficient in terms of time and in terms of cost. And we're still having this discussion with the HTA at the moment about that. They were very positive, and now they've realised they were maybe too positive and it has to go to a stage 2 funding board, which is reasonable, I suppose. So briefly, the last 5 or so minutes of my talk, I'm going to talk about a couple of next things. ROSSINI platform. So I alluded to this at the beginning. There's loads of poor evidence. There's lots of guidelines about wound infection. It is a massive problem. We all recognise it. The issue is this evidence base is full of trials at very high risk of bias. And the system for assessing that is based on really old-fashioned contamination strata. This 1964, someone came up with this idea of wounds being clean or being contaminated or dirty or contaminated. And that lumps together any operation where you open a viscus into one group, the clean contaminated group, which means in reality, an operation on your neck or your lung or your gallbladder or your colon or your uterus are all the same. And if there's one really good trial in neck surgery, it gets extrapolated by the guidelines to apply to everything else. Similarly, a toenail operation and neurosurgery are in the same clean group because they don't open viscous. That means that the guidelines are really poor. We know that the existing evidence around how infections are caused, what the pathogenic organisms are, would suggest that that's a really silly way of making recommendations, and we'd like to do something about it. The ROSSINI platform aims to replicate ROSSINI 2 across different parts of the body. So it's all abdominal surgery so far, but we'd like to expand it and learn from different parts of the body in what is called a novel, it's called a basket MAMS design, which is there. So this is ROSSINI's arrow diagram flipped on its side, replicated 6 times to be done in breast surgery and groin surgery and cardiac surgery, neuro amputations, leg and thoracics. And these are separate trials run by their own CI to bring through some new talents, overseen by the national leads for their clinical area. And people think surgery is surgery, but actually there's 15 very distinct branches of surgery in the UK, and we very rarely meet each other physically. We have our own research teams, our own hospitals sometimes. So we are really expanding what we're doing across the body under one umbrella platform, kind of overarching management team. And this is that generic protocol that Max mentioned with the sub-protocols to different parts of the body. The really sexy bit that I think is exciting is taking what works in one part of the body, be it a single intervention or a combination. Say we find something that works in breast surgery and we say to the cardiac surgeons, that's geographically close, it's probable that it might work. Why don't we try this in your part of the body? Because it's likely the evidence base has been separate until that point. And similarly, we might just say, well, it works in leg amputations, let's try it in neurosurgery, just because I may be wrong and actually all the causes are all the same. But something that works in one part, that's the green arrows. The other part that we've taken to the funder, which I think is really novel, which we're still discussing, is adaptive randomization for an arm that looks like it's a winner to randomize deliberately 8, 10, 15 to 1 into it to prove it works quickly, or if it looks like a loser, again, over-randomized to drop it quickly. which I think is good. We're still in the design discussions. That's really complicated. I spent a weekend writing that list of problems, which you can't read from the back. It's just to define that there are a load of issues with even putting in a funding application for that. If you were on the funding board, I can imagine what you'd think. We're very lucky that the HTA has said they want to fund platform trials, as you'll be aware. Lord Bethell stood up in the Houses of Lords 2021 and said we want another 10 recovery trials next year because the recovery trial was so successful. That's where I think a lot of this has come from with EME and HTA. We were lucky to get one of these application accelerator awards that allows us to meet and to start to try and answer these questions in order that by November, when the application comes out, we can put something in that has at least dealt with some of these major issues about how you do this, how you operationalize some of the stuff from a governance and a stats and a PPI and a clinical point of view. We've already had loads of meetings. We've had a kickoff meeting. The clinical teams have had meetings. We've had 3 of our PPI groups and the different pillars have met. It's going really well, and I'm very excited in a couple of weeks, 2 weeks today, in fact, 2 weeks yesterday, to be coming down to MRC with our big team of 26 co-applicants to thrash out some of the other finer points about how we do that. Final topic is Rossini Global. We would like to, well, firstly, we're going to change its name. There's too many Rossinis already. So it's now called Marlin as of last week. We'd like to do what we've done in the abdomen in Rossini on an international scale using that global network that delivered Cheetah and Falcon so well. We'd like to do something a bit more novel with that group. And that's really what this proposal is. We've got these 7 hubs who are core-funded by the UK government, effectively. We've had £14 million to do the global surgery unit, so they're staffed and ready, and they've proved that they are more than capable of delivering trials. Our issue is that different interventions are sometimes not allowed in certain parts of the world. So they don't have approvals and they simply won't get them. Or there are equipoise and kind of practice issues that means that some of the things for wound infection are inappropriate. I've discovered in sub-Saharan Africa, in some countries, the ritualistic washing before an operation means that our proposed intervention to introduce a chemical-based wash before surgery would not be acceptable or would be so biased it wouldn't work. So, plus, getting through multiple ethics boards across the world is a challenge. So trying to do a MAMS trial where everything starts on day 1 is an issue. We've divided this up into kind of 3 phases, and we've picked 3 sample interventions you can see on the right. We've said, to make it really simple and understandable across the world, we'll have some form of pre-op intervention, some form of intra-op intervention, some form of post-op intervention. And we'll write a really nicely vague protocol that allows us to swap new ones in, a kind of A2 and an A3 as time goes on. And we will randomize patients initially on a 1-to-1 basis. And the DAC wound dressing, that company is a lovely company. They're very interested, and we've already submitted to ethics for a 1-to-1 trial in 2 countries on a protocol that is going to be based on an expansion into a full-on MAMS. I think there are some questions around how we do this, how we pool the control arms. It's a kind of modular MAMS, I'm calling it in my mind, but I bet Max has got a better name for it. And then whether we can have, if one country can do all three interventions, whether we can just do a more of a Rossini 2 style study and how we cope with that opening in a stage-wise fashion. Final slide. The MAMS trial is brilliant. Thank you very much. I think we are using it in a pretty good clinical example in wound infection. We haven't solved the problem yet, but we're going to. I'm convinced that the practical design this afternoon is going to be where we are in wounds in 5 or 10 years' time as we start to look at people's microbiome in more detail on the skin and on their insides. We'll be able to select interventions for patients on a personalised way. So I think that's probably also where we are heading. And I'll stop there. Thank you.
Thank you very much indeed. I don't know if there's any one question on fact, but can't see any. So I think thank you very much indeed, Tim. Thank you. So I think we'll move forward